Hello, I'm Sean Gibbons, an associate professor at the Institute for Systems Biology, and our lab studies the human microbiome and how it affects human health and disease. And today I'm here to talk about our latest paper, which is about to come out online in the journal Nature Microbiology. This work was driven by Dr. Nick Quinn Bowman, a PhD student in the lab who recently graduated and got his PhD, and also with my co-senior author, Christian Diener, who's now assistant professor at the Medical University of Graz in Austria. So to give a little background on this paper, which is titled Microbial Community Scale Metabolic Modeling Predicts Personalized Short-Chain Fatty Acid Production Profiles in the Human Gut. It's a bit of a mouthful, but basically it's an implementation of our metabolic modeling tool set that we've been building for the last several years, pioneered by Dr. Christian Diener, who I mentioned earlier. It's a method for simulating the metabolic outputs of an entire microbial community given a particular dietary input to a human host. And in this particular paper, Nick is trying to point this modeling framework at a really important question. You feed different people the same exact food, and you get different things coming out the other end. There's a lot of heterogeneity in how people respond to diet. And he focused on a particular set of molecules. So with all that as background, I thought I'd turn it over to Nick to give us an overview of the paper, why this particular problem was chosen, and why short-chain fatty acids. Nick.
Thanks, John, for that great introduction. And yeah, as you alluded to, for this project, we were really looking to determine whether or not we could use this modeling platform to predict functional metabolic outputs from the gut microbiome on an individual specific level. In this case, we were specifically looking at short-chain fatty acids, or SCFAs, which are a set of molecules produced by the microbiome that have a number of really important functions in the host. They help to keep the lining of our gut healthy. They help to improve insulin resistance, sort of preventing the onset of prediabetes and diabetes. They're important for the gut-brain axis, helping to keep our brain healthy and prevent neurodegeneration. And finally, but very importantly, they're also important in modulating immune response. So through the modulation of different immune cells, they can actually tamp down an inflammatory immune response and bring the body back into a sort of less inflammatory state. And this is important for preventing the onset of certain diseases like inflammatory bowel and Crohn's disease, certain cancers, and other cardiometabolic diseases. In this case, we were really interested in determining whether on an individual specific level, our model was able to predict outcomes. Since, as Sean alluded to, given the exact same dietary input, the gut microbiome and differences in the gut microbiome between individuals leads to very different outcomes in terms of the production of short-chain fatty acids, as well as many other metabolites.
And these short chain fatty acids are kind of ephemeral in the body, right? They're produced quickly, but they're also consumed quickly. It makes them very hard to measure. So at the end of the day, how did you go about validating that the model predictions were correct?
It's a great question, and it's one of the primary challenges that we faced when constructing this project. In the body, it's very difficult to measure SCFAs, and that's because as soon as they're produced by the microbiome, they're just as quickly consumed by the cells in our colon. So not very high levels of these SCFAs actually reaches circulation where they can be measured. So to validate the predictions of our model, we actually turned to communities outside the body. So either in vitro cultures of lab strains put together, or cultures of fecal samples collected directly from study participants that were then cultured anaerobically. These sort of model communities allowed us two things. We could measure directly the composition of the microbiome, so which microbes are present in each community, as well as, without interference from the host, measure the production of short-chain fatty acids. So without the cells in our colon quickly consuming these metabolites, we could measure how much of each metabolite was produced by each individual community.
And we show in the paper that one can predict these short chain fatty acids relatively accurately, not perfectly, but relatively accurately. So then, once you have this capacity to make these personalized dietary output predictions based on the microbiome, what do you do with that? What kind of phenotypes is that affecting in the host, and how does one go about validating those effects? What did you do in the paper?
We were very interested in exactly that. So after we showed that our predictions were relatively accurate on an individual specific level, we next turned to a cohort where we had thousands of different individuals. And what we saw after building models for each of these individuals and simulating their growth, we saw that predicted levels of butyrate specifically showed very strong negative correlations with several markers for poor cardiometabolic health. So essentially, individuals that had poor cardiometabolic health were predicted to produce less butyrate. Some of these markers included C-reactive protein, which is a measure of sort of systemic inflammation, LDL cholesterol, which is often referred to as sort of bad cholesterol that can lead to the onset of cardiovascular disease. Insulin resistance, measures of insulin resistance that can lead to prediabetes and diabetes, as well as several others. So not only did we see sort of a quantitative agreement between our predictions and measurements of SCFA production, in this case, we also saw sort of a qualitative agreement between predicted levels of butyrate production and sort of the physiological effect that we would expect butyrate to have in the body.
That's great. And there were limitations to the way we were able to implement these models. So I thought maybe we'd briefly brush on maybe one of the more important limitations, which is the diet. So what choices did you make in the current paper in how you model diet? And what might we do in the future if we wanted to make these models perhaps more predictive?
Absolutely. So one of the primary inputs into these models is the diet, which sort of constrains the model on which dietary precursors it can use for growth. The difficulty here is that we didn't have specific data on what each individual in our study was eating. So in this case, we had to sort of assume a standard European diet for each individual and apply that broadly across all individuals, which isn't necessarily relevant since every person eats something slightly different. Excitingly, other work out of our lab has been working towards fixing this problem. Essentially, it uses metagenomic data to map to a database of known foods, which we can then work backwards and use that to build sort of an individual specific medium of what that person was eating, and apply that to our metabolic models to help improve the predictions that come out of our modeling framework.
All right. So that's one way we can make these models better. So assuming that the modeling is working pretty well, and we have this capacity to sort of simulate dietary inputs to a given person's model and see how they respond in particular, how do you envision the implementation of these models and the translation of these models into the real world? Where are we going with all of this?
I think that's the most exciting part of this work is that it's sort of ongoing and it has a lot of promise. So yeah, one of the things that we sort of see moving forward, one of the benefits of this modeling framework is that it allows us to test all sorts of different interventions, whether they be complete dietary switches or sort of the addition of prebiotic fibers or probiotics, sort of, putatively healthy bacteria that we can add to an individual microbiome. Um, and it allows us to test all of these, individually as well as in combination. And so what we showed in the paper is that we can use this framework to sort of find an optimal intervention aimed at increasing SCFA production for an individual person, given their unique sort of compositional context. And so moving forward, I think this could serve as a powerful tool in precision nutrition, sort of building optimal interventions aimed at improving metabolic outcomes from the gut microbiome for each individual person.
That's great. Thanks, Nick. I think this is an exciting time in the microbiome field where we can start to map this ecological composition to these functional outputs. And you mentioned precision nutrition or personalized nutrition. So obviously this model has applications there, but we might actually even say precision medicine in the sense that butyrate as a molecule is as potent as many drugs that are on the market in terms of improving your cardiometabolic health and reducing inflammation. So hopefully in this next few years we're proving Hippocrates right that food is medicine. It's just not everyone needs the same medicine, right? That different foods apply to different people to get the same drug output, and that's where we're going with all this. All right. Well, yeah, I'm so excited for this paper to come out, and great work and congrats.
Thank you so much, Sean. I'm very excited.